So you might actually be losing marks on the fluid mosaic model, and I promise you, it's not because you don't know the biology. It's usually because of one tiny, totally avoidable mistake in your phrasing. Welcome to this explainer. Look, we are skipping the fluff today. We're diving straight into exactly how to master the fluid mosaic model for your Cambridge A-level biology exams. Because if you want that A-star, you really need to understand exactly how the examiners think, and more importantly, how they grade your specific wording.
Okay, take the phrase, phospholipids move around. Now this is a flawed B-grade student answer. Now contrast it with this one. Phospholipids demonstrate lateral movement within the bilayer. Boom, that is your top band A-star answer. Instantly, you can see how a vague description of movement is literally costing you critical grade boundaries. Now, the underlying biology in your head might be exactly the same for both, right? But the terminology you put on the paper, that sets the stakes. You absolutely have to be precise.
And honestly, this brilliantly illustrates a core truth about these exams. The difference between a B and an A-star is analytical depth, not just superficial breadth. Rote memorization alone, it just won't cut it. Examiners aren't looking for human flashcards. They are actively hunting for logical, structured scientific reasoning. The Cambridge assessment prioritizes how deep you can analyze a concept, how well you understand cause and effect, and your ability to connect this with totally different areas of the syllabus. Making those cross-topic connections is what catapults you into that top tier.
All right, let's dive into the foundational concept itself. The fluid mosaic model is our conceptual framework here. Think of it kind of like a microscopic pool of oil, where the phospholipid bilayer is the fluid, and diverse proteins are just floating around in it like a dynamic, shifting mosaic. Now, what's really interesting about this is what the principal examiner reports explicitly tell us. They say top candidates always define these key terms before using them in extended responses. It establishes your knowledge right out of the gate, so never assume the examiner knows what you mean. Prove it to them by defining your terms clearly.
Which brings us to the ultimate A-star answer framework. There are 3 super clear steps to follow here: concept, evidence, and reasoning. Let's say the question is about temperature affecting the membrane. Step 1 is stating the concept, that higher kinetic energy increases fluidity. Step 2, pull the specific data points straight from the graph provided in your exam paper. And step 3 is the scientific reasoning. This is where you explain how that increased kinetic energy actually disrupts the weak hydrophobic interactions between the phospholipid tails. Connecting these 3 elements literally guarantees marks because it perfectly aligns with exactly what examiners are scanning for.
But here is a vital question for you to chew on. What is the single hidden examiner clue that changes your answer completely? In just a moment, I'm going to show you exactly what this clue is and how it totally flips your whole exam approach. But first, you really need to master the procedural framework that makes spotting that clue completely obvious in the first place. And that framework is what we call the method mark strategy. Let's just walk through this standardized sequence together. When you're tackling those massive, complex past paper problems, you must follow these exact steps. First, deconstruct the question to identify the key elements. Like, what are they actually asking you to do? Second, extract relevant data from the stimulus materials. Pull those exact numbers from the tables or graphs. Third, select your analytical framework. Fourth, execute the analysis with clear notation. Always show you're working. And finally, present your conclusion using the correct terminology. This step-by-step process is exactly how process marks, those highly valuable method marks, are awarded, even if your final answer turns out to be slightly off.
Now, even with a brilliant process, students still stumble under pressure. It happens to the best of us. There are 3 painfully common mistakes pulled directly from the Cambridge Principal Examiner reports. These are the exact spots where really smart students unnecessarily bleed marks on the fluid mosaic model and related calculations. Let's look at exactly what those mistakes are. Number 1, forgetting to include units in calculations. I mean, we've all been there, panicking and leaving a number completely naked on the page. Number 2, failing to double-check those units and the associated terminology. And number 3, providing answers that just aren't specific to the given context. I have to strongly warn you about those first 2. Examiners consistently note how easily marks are lost simply by missing units when you are calculating things like membrane thickness or magnification from a micrograph. It is a devastating and totally avoidable way to drop a whole grade boundary. So to combat that terminology confusion, here is a highly effective, totally practical memory hack. Create a mnemonic using the first letters of key membrane terms. We're talking about phospholipids, which form the main barrier, glycoproteins, used for cell recognition, cholesterol, which regulates membrane fluidity, and channel proteins, acting as selective gates. Go ahead and create a memorable, maybe even a funny sentence out of those first letters right now. Under intense exam pressure, a simple mnemonic ensures you never confuse the complex terminology and you always have the right vocabulary ready to deploy when you are evaluating membrane structures.
All right, now it's time for a quick interactive challenge. I really want you to actively engage with the material we're covering. I'm going to describe 3 data scenarios. Your job is to pause this explainer and predict the correct statistical test for each dataset. Data interpretation is a totally critical experimental skill for this syllabus, so let's test your judgment right now.
Imagine 3 columns representing different types of experimental data you might extract from a biological investigation. Column 1 is comparing means, column 2 is categorical data, and column 3 is relationships between variables. Mentally lock in which statistical test belongs in each column. Would you use a t-test, a chi-squared, or a correlation test? Take a second, actually think about it, and lock in your answers. Okay, got 'em? Let's reveal the correct answers. For comparing means, you absolutely must use the t-test. For categorical data, it's chi-squared. And for testing relationships between variables, you use correlation tests. The crucial point here is that selecting the right analytical framework is absolutely essential for handling and evaluating experimental skills. If you misidentify the test at the start, your entire statistical analysis falls apart, and you just lose all those evaluation marks. And that brings us to the absolute highest level of assessment. Just listen to this direct quote from the Examiner Insights. Top-scoring candidates demonstrate evaluative thinking by questioning assumptions, considering different perspectives, and justifying decisions. You have to push beyond basic problem solving. When you apply that deconstruct, solve, evaluate method we talked about, you must then check your data for reasonableness. You have to actively identify limitations in an experiment and evaluate alternative interpretations of membrane behavior. That level of critical analysis, that is exactly what secures those top descriptive and evaluative marks. To ensure you consistently hit those top mark bands and master literally every tricky concept Cambridge throws at you, make sure to subscribe to our YouTube channel, Ace the Exams, at acetheexams_IGCSE. We are absolutely here to set you up for continuous success in your exam preparation journey with these highly focused explainers.
Oh, and before we go, remember that hidden examiner clue I promised you earlier? Well, it all comes down to one final diagnostic question. You know the biology, but what happens when the exam command word completely changes the mark band you're graded in? You could have the perfect fluid mosaic model answer, but if you describe when the examiner told you to explain, you get a 0. In our next explainer, we reveal the exact Cambridge command words that dictate your grade and the hidden rules you must follow for each. Click on it right now, because as you've seen today, understanding the biology is only half the battle.